This is Bio 181 video from chapter 15. And in this chapter, we're looking at transcription and translation. This is described, these processes are described by the central dogma of molecular genetics. And in this dogma, just a way to understand how information moves from being stored in the base pairs of DNA, so A, T, C's, and G's, into information that is stored in slightly different base pairs in another type of nucleic acid called RNA, specifically messenger RNA. And then that information is taken and used to make proteins. So the two main processes we'll learn about are transcription. So transcription occurs, CR, in the nucleus of a eukaryotic organism. Okay. And a prokaryotic organism like bacteria, it's just going to be in the cytoplasm, but we'll focus on eukaryotes. And then the messenger RNA is then going to leave the nucleus where the DNA stays there and remains protected. And the process of going to proteins is then called translation. I know these are very similar, so just pay attention so you don't get them mixed up. Translation is going to occur in the cytoplasm, and the messenger RNA is going to go find a ribosome. So ribosome, this is the site of protein synthesis. So messenger RNA, just a few things about it. It's a type of ribonucleic acid. So ribose is the sugar. If you remember the nucleotides, every monomer of a nucleotide has a sugar, a pentose, which means it has 5 carbons, and then it has a phosphate group, okay, and it also has a nitrogenous base, okay, so a nitrogenous base. So RNA is similar to DNA in this way. This is, again, the monomer, which is a nucleotide. And the difference here is that the nitrogenous bases are AU, so this is the different one, C and G. Okay. And it pairs, so whereas A previously would pair with thymine in DNA, A will pair with uracil, and cytosine will still pair with guanine. RNA is also single-stranded as opposed to DNA, which is double-stranded, a double helix. So it's single-stranded. It has a different sugar. The sugar is ribose. In DNA, it was deoxyribose, and the base pairs are different. Okay. And its purpose is to bring the information from DNA to the ribosome. So the genetic code for amino acids, which are the building blocks of proteins, right? These are the monomer of proteins. So those are the things that we put together, covalently bond together to make a protein. And it turns out that the code that specifies these in terms of the DNA and RNA is the same for almost all organisms, right? So basically all organisms, which is really cool. There's a few differences here and there, but you can take the DNA out of bacteria and then put it into a different plant and create a transgenic corn that makes a bacterial protein because you give it the same DNA. So this is showing the process of going from DNA. You've got a 3-letter sequence of your DNA. It's going to be transcribed into a 3-letter sequence of RNA, and then that is going to be translated into amino acids. So again, this process is transcription. Okay. And this process from RNA to the protein chain is translation. All right. Just a few vocabulary terms for the last slide here in this video. The genome, this is all of, so genome is your complete set of genes and all of the other DNA. So this is all the DNA in an organism. You can think G for genes. The transcriptome, that's Okay, so this is all RNA transcripts. So that's going to be all of our messenger RNA and in an organism. And then the proteome is all of the proteins in an organism. So there are studies that look at all of these, so all the proteins. The DNA is important to know what's possible, right? That's your entire cookbook. The transcriptome is looking at what's actually being copied out. So it's just a subset of that that's actually being made by a cell. So you're going to have different things made in a liver cell than you will in a brain cell or you will in a muscle cell. So it's often really helpful to look at the transcriptome in an entire organism and also at the cellular level. And then the proteome is looking at all the proteins that are actually made. So there can be, actually, we'll talk about this, but the DNA can be put together in different ways to create variable transcripts, which then can create variable proteins. All right, that's it for this video.